Let's get work on transcription. So we went through the sort of the basics of transcription or the basics of gene expression, transcription and translation on Monday. Today we're going to get in the actual process of transcription, all right, the first step of gene expression, all right. We know transcription is producing what? RNA. What type of RNA? mRNA. Okay? The overall end result of transcription is a complete, processed, ready to be translated RNA. Okay? So we have transcription, which is going to produce part of really the precursor. All right? We're going to produce that nascent RNA, a pre-mRNA, or a primary transcript, whichever word you want to say it is. Okay? And then that's going to be processed, and then we'll export the nucleus to the cytoplasm or the rough endoplasmic reticulum to be translated. Okay? That's the overall process. Really, there's two steps. There's the transcription, and then there's the RNA processing. Okay? So, what does this? We're going to talk about the proteins that are involved in making an RNA transcript, a primary RNA transcript. All right? And basically, there's one big player. Okay? There's one player that's there all the time, and that's RNA polymerase. DNA polymerase does what? Synthesizes DNA off of what? A template strand. So RNA polymerase sounds pretty much the same thing, right? Does what? Synthesizes RNA from a DNA template. Okay? Difference between the two. What does DNA polymerase require? A primer. Okay? DNA polymerase requires a primer. RNA polymerase does not. Okay? It will synthesize new strands on its own. It's able to assemble polynucleotides in a 5' to 3' direction, right, without the use of a primer. Okay? So, that's the main player, RNA polymerase. And there's a couple different RNA polymerases depending upon what type of cell you're looking at, right? Humans have 3 different RNA polymerases, right? One RNA polymerase 1, 2, and 3, based upon when they were discovered. Okay, polymerase 2 is the one that is mainly important for making messenger RNA. Okay, messenger RNA. What other types of RNA are there? Transfer RNA, which is involved in translation. And another type of RNA is ribosomal RNA. Okay? If RNA polymerase 2 is important in making messenger RNA, what do RNA polymerase 1 and RNA polymerase 3 do? Think about that. What do you think they do? They make the other 2 types of RNA. Okay? So RNA polymerase 2 is mostly used for making messenger RNA, and RNA polymerase 1, Pol1, and Pol3 are used for making ribosomal and transfer RNAs. Okay? So this is the overall process of transcription. Okay? RNA polymerase 2 is the big player. And it's important that an RNA is made at a certain point. It begins at a certain start point, right? We went over the codons on Monday, right? And we talked about reading frame. If the RNA polymerase is off a base, it doesn't mean, the code doesn't mean the same thing, right? So it's very important at getting the RNA polymerase at the right start point. Okay? So we have, cells have evolved very specific ways to attract RNA polymerase to the right start point. Okay? And that is the process of transcription called initiation. Right? It's a way of recruiting RNA polymerase to the right point to start. Okay? And so transcription first involves initiation, recruiting RNA polymerase. Right? Then it starts producing an RNA, then it elongates along the chain. Right? So then elongation, and then there has to be a way of stopping it in termination. All right? So we have three distinct steps in transcription: initiation, elongation, and termination. All right? Initiation is getting RNA polymerase to the right start point. Okay? So here you have a transcription unit. All right? What you see up there in dark blue is the coding region of the gene. That's the part of the gene that is going to be or lead to a protein. Okay? You have a promoter region. All right? A promoter is a region of the DNA that helps to regulate the expression of the gene of interest. Okay? So that promoter region is usually not, quote unquote, transcribed. It doesn't turn over, is not part of an RNA. Okay? But there are distinct sequences within the promoter region that proteins bind to that will help recruit RNA polymerase to the right start point. Okay? And there are thousands of different promoters in your body, right? And they're all responsive to different other proteins, right? And other DNA binding proteins. Okay? Simple explanation of a promoter. All right? You understand what a promoter is? Yes? Okay. So, you have initiation. This is showing, up here is showing that you have RNA polymerase comes in and the unwound DNA, and then you have an RNA transcript that begins. It's a very simplified version. There's plenty of proteins that are there that are not shown here. Okay? So this is just showing RNA polymerase. You see it begins to make a transcript, right? It processes down the DNA template, producing an RNA, a complementary RNA, right, off the DNA template strand. And then at the end, you get termination. It somehow disassociates meaning it separates from the DNA, and then you have the polymerase, you have the completed RNA transcript, and then you have your double-stranded DNA that all kind of go their separate ways. Okay? It's the simplified version of transcription. Right? It's not that simple. All right? So we talked about the promoter. Okay? That helps to promote gene expression or RNA synthesis. Okay? And at the end of the sequence, there's what we call a terminator sequence. It's a certain sequence of DNA that makes the RNA polymerase less attracted to the DNA. Right? It disrupts the binding of RNA polymerase to the DNA template. Right? When that's there, then it forces everything apart. Okay? And so you have this whole stretch of promoter, gene, and terminator sequence is your transcriptional unit. Okay? So you have your three stages of transcription. We're going to talk about initiation. All right? So we have promoters. Promoters are important in placing the RNA polymerase at the right start point. Okay? Get it lined up correctly, okay, so that it can do its job, right, and produce the right the right sequence. All right? So promoters signal at the start point and usually extend several dozen nucleotide pairs upstream of the start point. So we tend to, us scientists tend to draw it something like this. Right? So if this is your promoter region, this is your transcription start point, usually an arrow, and then we have your 5' terminator sequence. Right? So this would be your promoter. Okay? And there are certain elements within a promoter which are important. All right? So promoter is basically anything upstream of the start point that helps regulate the expression of this gene. Okay? Example being the myosin specific, a muscle specific promoter that is upstream of the myosin gene, which is myosins found in skeletal muscle. Okay? And there are thousands of other promoters. There are sequences within the promoter region that DNA binding proteins recognize, that RNA polymerase recognizes, okay, that will help to kind of align where it is. Right? And then we'll go through a couple of them. All right? So you have basically those proteins that bind to the promoter help recruit RNA polymerase II to the right start point, to the right point to begin, and help to stabilize the interaction between RNA polymerase and the promoter. Okay? Okay? That all together, the RNA polymerase and whatever proteins are involved that help to stabilize that complex form what's called the transcription initiation complex. All right, so it's not just RNA polymerase that's there. There are other proteins that are involved in starting transcription. Okay? And then this says a promoter called a Tata box. It's not. It's sort of a wrong nomenclature because really within a promoter sequence, okay, so this whole thing is the promoter, okay? Within this promoter will be a sequence of DNA that's TA, right? What we call a Tata box. It's a short stretch of DNA nucleotides on the DNA strand that's ATA, something like that. It has a stretch of A's and T's. Right? We call it a Tata box, and that is important for binding of RNA polymerase to that area. Okay? And it's conserved over genes. So if you compare the promoter region for a gene in monkeys, in humans, in yeast, in worms, in Drosophila. If you compare the promoter regions for conserved genes, they have similar structure, and that Tata box is sort of spaced along the same spacing prior to the start site across species. Okay? So it's very important in the lineup of RNA polymerase to the transcription start site. All right? So we're still in. Here's your Tata box. All right? So you have TA, TAA, right? Whatever it might be. Upstream of the start point. And then you have your template strand. That's a eukaryotic promoter. All right? Certain transcription factors, whatever they might be. There are thousands, on the order of 10 to 20,000 different transcription factors. Okay? Couple of them that you may be familiar with. Estrogen receptor. Have you heard what estrogen receptor, you know what estrogen receptor is, right? What it's involved in. Yes? No? Maybe? Have you heard of an estrogen receptor before? Yes? No? How many of you heard of breast cancer? How many of you heard of hormone responsive breast cancer? Okay? So hormone responsive breast cancer means that those cancer cells produce estrogen receptor, and they respond to estrogen fluctuations in estrogen hormone. You all know what estrogen is, right? Okay. So the way estrogen works in promoting cell growth, right? Estrogen's a lipid. It's a steroid. It's a lipid. It crosses the cell membrane and binds to the estrogen receptor that's floating around in the cytoplasm. Okay? When estrogen binds to the estrogen receptor moves to the nucleus and actually is one of these transcription factors. It works in that way. Okay? Binds to a particular element within a promoter and helps recruit transcription of RNA polymerase II to a gene, which then is transcribed. Okay? So now, hormone-responsive cancers are going to produce estrogen-responsive genes because they have the receptor present, the hormones present. That's going to influence gene transcription. Okay? Now, that's one example of a transcription factor. There are loads and loads and loads of others. Okay? That transcription factor recruits RNA polymerase II. RNA polymerase II binds and then unwinds the DNA, and you get production of an RNA from that promoter sequence. Okay? Abby.
What's the difference between a transcription factor and a promoter?
So, a transcription factor is a protein that will bind to a promoter region. A promoter is a stretch of the DNA. Okay? So, a promoter is a DNA sequence. It is TA, GG, whatever it might be. All right, CG, all the way, whatever. And the transcription factors will recognize certain sequences on the DNA and bind to the DNA in order to produce this transcription initiation complex. All right, other questions? Good question. All right, so that's initiation. It is ways of the cell to recruit RNA polymerase to the right spot. And the spacing is conserved over species, and transcription factors or other proteins beside RNA polymerase II help recruit, bless you by the way, help recruit RNA polymerase. Questions on initiation? Okay, so this is all getting it started. Now it begins. RNA polymerase reads, binds to the DNA, template strand, recognizes what base is there, and then brings in the correct ribonucleotide. All right? And it will unwind the DNA, right? Recognize, and then bring in, excuse me, then can bring in the right nucleotide and produce the transcript. All right? So transcription progresses at a rate of about 40 nucleotides per second in eukaryotes. Do you believe that transcription rate will be faster or slower in prokaryotes than in eukaryotes? Why? Why faster? Sarah?
Is there less DNA because prokaryotes are more simple?
Not less DNA. We're still saying that we're still transcribing a similar size. There's no.
Does it have to leave the nucleus?
I'm just talking about RNA polymerase is bound to a DNA, and now it's going to go 40 nucleotides per second in eukaryotes. How do you think it's going to be faster in prokaryotes? Is it going to go faster? Yes or no? You say yes. Why? Yeah, they don't have the proteins that are bound to the DNA. Right? There's less other stuff in the way. Right? So it can process faster. Okay? So here's sort of elongation. Elongation's not hard. Nucleotides brought in based off of the template strand, Okay, you get synthesis of the RNA single-stranded molecule based off the template, and then DNA reattaches, right, rewinds. The structure of the RNA polymerase itself, the way it fits and binds to RNA, binds to the DNA, actually helps to unwind it. There's no helicase that's needed here to unwind above or before the enzyme. It does it itself. And it's the way that the enzyme is structured and how it binds to DNA. There's a portion of the protein that actually works as a rudder, such as you have the two strands of DNA, and RNA polymerase sort of binds here and pushes the two strands apart. Okay, and then works down that way. All right. Questions on elongation? All right. Termination. So we have this million base pair stretch of DNA. Okay? And we're particularly want one, say, a couple thousand base pairs of that million base pair stretch of DNA to actually is part of your gene. That's what you want to be transcribed, right? So how does a cell prevent RNA polymerase from starting at one point and not ending at the end of the double strand and going transcribing, say, 990,000 more base pairs than it needs to? This is where termination comes in. There are sequences built into the DNA, all right, that will alter the way RNA polymerase is attached to the DNA, and then it will cause it to separate. These are terminator sequences, all right? And this is different in bacteria and eukaryotes, all right? In bacteria, all right, termination occurs based upon the sequence, and then that's it, all right? In eukaryotes, you end up with this termination sequence, and then it keeps going a little bit for about 10 to 35 nucleotides. All right, so there's a slighter extension beyond the terminator sequence. All right. The terminator sequence is called the polyadenylation sequence. What does polyadenylation mean? What does poly mean? Many. All right, so what does adenylate or adenylation sound like? What's that? What is that? Adenine. Many adenines. Polyadenylation. Okay? So we're going to talk about two different polyadenylation sequences or polyadenylation that's important in this. All right? So a terminator sequence in the DNA has a run of adenines, okay, at the end. That, when an RNA polymerase goes down and reaches that point, so it looks something like this, right? So we have, here's your DNA, okay, da-da-da, then it reaches this point where it's A, on the DNA, okay? When that occurs, RNA polymerase we'll make a transcript, right, beyond this, and then we'll keep going, say, 10 to 35 nucleotides beyond that. When that occurs, then, once it gets beyond that, it separates off, right, DNA goes back to being double-stranded, and now you have your primary transcript has been produced. Okay? So it goes sort of beyond that point. Questions on the polyadenylation sequence? It is part, so you have a terminator sequence that is part of the DNA sequence, okay? That signals to the RNA polymerase. That's the end of the gene, okay? So now, at the end of the terminator sequence, we have this primary transcript, okay? Here's what your primary transcript looks like. All right, 5 prime, 3 prime. There has a bunch of T's in here. Right, that's the end of the transcript. And then there's, say this point right here is probably your AUG, your start codon. There's usually a 5 prime portion that's not part of the actual gene. All right? So there's this long piece. All right? What's inside there now, what is going to happen in eukaryotes is now this RNA, it's not ready to leave the nucleus yet. Right? It's not ready to be translated. There's a couple processing steps that have to be performed for it to leave the nucleus and to be readily translated into a protein. Okay? So this is RNA processing. DNA itself, and every gene you have, has a bunch of junk in it. Stuff that doesn't end up part of the protein. Okay? But it is transcribed as part of the RNA. All right? So we have really 2 different parts of the DNA. All right? So if this is your RNA, here's your DNA. All right? And say this part right here is part of the will end up becoming the protein, as is this part and this part. Okay? In the rectangles up there, that's what we call exons. That's what's going to become, which will become part of, which codes for the protein. Okay? That's the important part. There's stuff in between that's important, but it's not important for the function or the production of the protein of this gene. Okay? So we have exons in rectangles and your introns in between. Right? Intronic DNA does not translate over to a protein. Okay? RNA polymerase doesn't recognize intronic versus exonic DNA, or what's going to become protein or what's not going to become protein. It just produces an RNA transcript off of all the RNA. All right? It goes from start point to termination point. Doesn't care what's in the middle. All right? But part of the RNA processing is to remove the chunks of DNA or chunks of RNA that don't code for a protein. All right? So it's going to take this exon 1, 2, and 3 and splice them together. Okay? Because on the RNA you have exons 1, 2, and 3. Right? And what you want is to have something like this. Well, 1, 2, and 3. You want those exons to come together because now in translation, when translation occurs, we don't want to translate intronic DNA. That's going to throw off our reading frame, right? And we'll end up with the wrong protein. So this is splicing, right? So we have really 3 things that are going to occur in RNA processing. We're going to give the RNA a signal, or that's so that it can exit the nucleus. All right? And there's two signals that will help for translation. Right? We're going to modify the RNA in such a way that they can leave the nucleus, be translated, and then finally we're going to remove the introns. We're going to remove the junk. All right? So each end of the pre-mRNA, the RNA transcript that is produced, right, is modified in a particular way. All right? So we have a 5' end and a 3' end. They're altered. These are the signals that are important for translation to occur, all right, and to protect the RNA from breakdown. All right? The 5' end is modified in that it adds what's called a 5-methyl cap. All right, a 5' methyl cap. So, how many of you taken organic chemistry? Nick, Dina, what's a methyl group?
One carbon.
What's that, Dina?
I'm not taking it now.
Nick, what's a methyl group? CH3. All right, so CH3. What's 5' mean? It's on the 5' end of a carbon, right? And we call it a 5' methyl G because it's on a guanine. Okay? So this 5' methyl group is going to be added to the end, the 5' end of the RNA. All right, so the cap. The purpose of the cap is that ribosomes will recognize, this aids in ribosomal binding to the RNA. All right, so now you put on the 5' end, not the 3' end, because if ribosomes are going to bind, you want to delineate this is the right end to start at, not the other side. Okay? This aids in binding. Over here, make it bigger. All right? Move that out of the way. The 3' end gets what's called the poly A tail. What's poly A? Polyadenylation. All right, so here we have, this is going to be here's our RNA now. Okay? Our pre-mRNA still has, right, these are your exons, still has introns. We're going to add your 5' cap, right? And then at the end of it, we're going to add a string of adenylation. All right? What do these modifications do? They allow for export to occur. Without these, the RNA doesn't get out of the nucleus. Okay? It allows for ribosomal binding. All right? So it helps ribosomes to attach to the 5' end. And the poly A sequence at the end actually signifies that this is a messenger RNA and protects it from enzymes in the cytoplasm that chew up RNA. All right? The enzymes that are in cytoplasm that chew up RNA are meant as an antiviral mechanism. Viral genomes can be RNA. RNA goes in, your cell recognizes that and chews it up, okay, to prevent from viral infection. But when an RNA that's made from you, a messenger RNA, has these things, it's protected. It gets translated. All right? It's a way of identifying, sort of a calling card that this is from you and is important in that way. Questions on that part of the processing? So 5' cap, poly A tail, and what they are used for. All right? We have to splice the RNA. All right? So splicing. Here's your transcript. Okay? This is your pre-spliced transcript. It has the 5' methyl cap, right? Modified guanine nucleotide. But here's your protein coding sequence, you have part of it is a UTR, which means untranslated region. All right, you have a 5' UTR, you have a 3' UTR, right? Untranslated regions. And then this is really the important part, your protein coding sequence. Here's your poly A tail, right? Could be from 50 to 250 adenines that are added, whatever it might be. Okay? That's sort of where we want to be. Okay? But inside this protein coding segment is still some junk DNA, or some, well, RNA that's coded from junk DNA. Right? And we want to splice that out. We want to take that out. We go through split genes and RNA splicing. All right? So we have non-coding regions of DNA, which are called introns. We need to get rid of the intronic RNA and put together, sort of link just the coding regions, just the exonic RNA. All right? So what splicing does, and what cells do when they go through RNA splicing, it removes the introns and joins together exons. Okay? So that when you're done, you have this stretch of RNA that only codes for the right protein. A ribosome can go through and read the whole thing. It doesn't have to decipher what's coding and what's not coding. Okay? What's junk and what's not junk. All right? You end up with a continuous coding sequence that translates over to protein. Okay? So here's a good representation of splicing. The dark red are your introns. Your exons. All right? That's the important parts. All right? So you have 1 to 30, 31 to 104, 105 to 146. Those are the base pairs, right, from the coding sequence of your RNA. We want to remove those introns. So a cell has developed very specific ways that recognize these intron ends, the boundaries. Okay? It recognizes the end, the end of an exon, the beginning of an intron. Okay? And will sort of take that out and link the two exons together. This is a property of the DNA sequence that's copied over into the RNA sequence. All right? That will be spliced out. You end up with a 5' UTR, which is important for binding of the ribosome. You have the protein, the coding segment, and then the 3' UTR, untranslated region, which is important in termination and separation of the ribosome from the RNA. All right? So that's sort of the nuts and bolts of splicing. All right? And say, this is the ideal, right? I want coding segment 1 to 146 that gives me a complete protein. But what if I spliced from this whole part out? So I end up with an RNA or coding segment that is 1 to 30, and then 105 to 105 to 146. I leave out that 31 to 104. Is that the same protein that's going to be produced? No. Is that protein going to have the same function? No. All right? So alternative splicing leads to different functions, leads to different proteins. And it actually helps to work in terms of evolution. All right? How does it occur? What's the importance of splicing? Why not just have exon all linked together, and then in the DNA, one coding sequence, so that you don't have to do this? So we have reading frame, and we have what's also called alternative splicing. Okay? Because if I go back here, giving one gene multiple functions and multiple proteins. Okay? So splicing is carried out by a group of proteins and RNA called spliceosomes, all right, which will recognize the intron-exon boundaries and cut them out. All right? And you have alternative splicing, which can alter sort of the sequence of exons and whether or not they're. This is what it looks like. So you have these RNAs and proteins that come together, and you see here there's a binding from between the small RNAs and the proteins that are sequence specific. So those four little dots, all right, those four little dotted lines represent base pairing between that small RNA and the intron. That's an intron boundary, all right? That delineates or signifies the boundaries. Recognize the boundaries, and then this whole complex comes together, bends the RNA, and then you get an enzymatic function that produces what's called this structure, which is called the lariat. That loops out, and then the two exons are brought together. You end up with your exons are spliced out, are spliced together. The lariat is cut out. So the intron's cut out, and you end up with the coding region together.
Do the introns go anywhere?
So the introns end up, the cutout lariat is RNA, but the cell doesn't have what? The introns are lacking two things. So the cutout introns are lacking two things. What are they lacking?
The poly A tail and the 5 cap.
They're lacking the poly A tail and the 5' cap. So the cell recognizes that as non-coding RNA and digests it. So these nucleotides will be cut up and then recycled to be used in another coding for RNA polymerase. All right? Questions on splicing? All right? And one other thing and look at splicing, right? You have your small RNAs that are binding, and that actually catalyzes splicing to occur. All right? So you have proteins that are involved, and you also have the small RNAs that function as ribozymes. Right? They have some type of catalytic activity and can, stimulate a process from occurring. So splicing, the 5' cap, the poly A tail, those three things have to occur in order for an RNA to be completely processed. Once that occurs, you have what's called a mature or complete messenger RNA. Right? That RNA now is ready to leave the nucleus, be exported, and bind a ribosome for translation. Questions on transcription and RNA processing? No? All right. So that will be the point where your quiz is up to. All right? And from last quiz to today. All right? If class is canceled on Friday, then your quiz will be on Wednesday. Regardless, your test is on Friday. Austin.
Okay, are you going to put up the lectures?
Yeah, the recordings? Yes. I'll put RNA can do. Right? So it was thought for a long period of time that messenger RNA and RNA is important in going from DNA to protein, and that's its main function, yes. But probably less than 2 decades ago, it was discovered that sometimes RNA has its own enzymatic function. It can function like an enzyme. It's not an enzyme, but it has some type of enzymatic properties, right? And we called this then a ribozyme. All right, so ribozymes, they sort of function as enzymes, but not in that the way the reaction they catalyze, lowers free energy, the classic, definition of an enzyme. It sort of starts a reaction from occurring. Base pairing occurs, and then you end up with a reaction that can occur. All right, so this occurs when you see that sort of introns can catalyze their own splicing. All right, so splicing can occur with or without proteins.